Okay, a very warm welcome to you all to this session on the topic of cornea. Okay, so we'll be talking about cornea today. So this is a topic from ophthalmology, and we are going to talk in about cornea in detail. It's going to be a very good lecture to you. I think I'll be giving you the enough amount of information that you need. So let's talk about the histology of the cornea. So whenever we talk about the cornea, it's very important for us to understand about the epithelial layer. Okay, this is the epithelium. Please draw with me. If you draw, you'll get it much more better. This is the stroma. This is the stroma. This is the epithelium. This is the epithelium. What you have here is the basement membrane. Okay, so if you zoom in the structure, the basement membrane is not actually the basal layer. Okay, the basal layer is something different. So this layer is basal layer. You have columnar cells which are in the lowermost area. You have this columnar cells. Okay, and then what you have is you have stratified squamous epithelium. Okay, so above 5 epithelium is stratified squamous epithelium, and then what you have is you have something which is known as non-keratinized stratified squamous epithelium. You have something which is known as non-keratinized stratified squamous epithelium, like this, non-keratinized stratified squamous epithelium. And what you have next is you have the microvilli. Okay, you have the microvilli. Okay, this is the microvilli. Okay, microvilli. So this is microvilli. This is stratified non-keratinized stratified squamous epithelium. And what is this? This is known as the wing cells. Okay, this is known as the wing cells, and this is known as this is known as the basal cells. Okay, the basal cells. And these basal cells are connected to each other by something which is known as hemidesmosomes. So they are connected by hemidesmosomes. So you might be thinking, why am I talking about these hemidesmosomes? Because these hemidesmosomes are important whenever I talk about corneal degeneration. So in corneal degeneration, these hemidesmosomes are very important. Now, let's talk about this microvilli. This microvilli, this is important for the attachment of the tear film of the tear film to the cornea. Okay, attachment of the tear film to the cornea okay, to the cornea. So you have the tear film. This tear film is very important for the lubrication of the eye. Okay, and this is done by your microvilli. Okay, and what you need to remember is, in corneal degeneration, corneal dystrophy, majorly corneal dystrophy. Okay, I won't say corneal degeneration, because corneal degeneration, whenever this term comes, you have to talk about the endothelial cells of the cornea. Okay, the endothelial cells of the cornea. Where is the endothelial cells? Here is the endothelial cells, and these endothelial cells are responsible for making the cornea in a dehydrated or a dry state. In this dry state, the cornea is non-edematous. It is non-edematous. Else the cornea will be fully hydrated, it will be fully edematous, and that will lead to defect in the vision. Okay, and the second layer we are talking about is the stroma. This stroma consists of glycosaminoglycans, collagens. Okay, and these glycosaminoglycans and collagen, which are very well arranged, are responsible for the transparency of the cornea. Okay, transparency of the cornea. Now what we need to remember is what type of collagen are present here. There is type 1 collagen in this structure, and in glycosaminoglycans, what you have is you have keratin sulfate, and you have chondroitin sulfate. Okay, chondroitin sulfate. Okay, so we are clear till here. Okay, now next extra layer which is added over is the Duas layer, and these Duas layer are present just above this Descemet's membrane. Okay, so this one over here, this line, okay, on which the endothelium rests, is the Descemet's membrane, and this is the endothelium, and this one is known as the Duas layer. Okay, this one is known as the Duas layer. Now this Descemet's membrane, it has two parts. Okay, this Descemet's membrane, this is all anatomy. Descemet's membrane they have two parts. One is a banded part, other one is known as the non-banded part. Okay, banded part and a non-banded part. Okay, whenever we talk about the banded part is already laid down in uterus. Okay, so it is present in utero. And non-banded part, this is secreted by the endothelial cells. This is secreted by the endothelial cells, and this is regenerative. Now the only regenerative structures that you have in the cornea are the non-banded part of the Descemet's membrane and the endothelium, and the epithelium. The endothelium, they lack the property of regeneration. That means whenever you have this endothelium, I hope you are getting this point very well clear, that whenever this endothelium are damaged or if they are traumatized, then this endothelium goes with the property of polymegathism and pleomorphism. So if this endothelium is damaged, then this endothelium, it tends to increase in size. Suppose I have the two endothelium and this endothelium is damaged. Okay, the preceding endothelium will increase in its size and change its morphology, which is known as polymegathism, and pleomorphism. Polymegathism and pleomorphism. Are we clear till here? We are clear till here. Now, next thing that is very, very important for us to understand, that if there is any break, in the Descemet's membrane, it leads to Habstria and it leads to Voxstria. Okay, it leads to Habstria and it leads to Voxstria. Okay, now what you need to remember is the physiology of the cornea. So physiology, I have already told you that it is dehydrated, it is avascular, and the major thing that you need to remember is the nutrition, the metabolism is aerobic. If you talk about the lens, it is anaerobic, but in this cornea, it is aerobic. And the nutrition, it comes from the aqueous humor. Now, what you need to remember is the endothelium, which is present like this, it has two mechanisms. One is the sodium potassium ATPase mechanism, by which the cornea is dehydrated. Just remember that the sodium is kicked out. So if the sodium is kicked out, the water will also be kicked out. So the water will not go inside the corneal endothelium, because of which there is no hydration in the cornea, leading to a normal cornea. If there is a hydration, it will lead to edema of the cornea, and we don't want that, right? The normal endothelial count, it's very important. The normal endothelial count is 2500 to 3000 cells per millimeter square. Remember this, okay? 2500 to 3000 millimeter square. Now, why is this cells per millimeter square? So why is this important? Because whenever we go through certain investigations and just check this endothelium, if it is less than 1500, less than 1000, then that won't be a suited, that cornea will not be suited enough for going under a keratoplasty. So that means basically I'm just taking out the diseased cornea and replacing it with healthy cornea. So if the cornea, it has only 1000 cells per millimeter square of endothelial cells, then I cannot take that cornea and replace it to someone. Okay, so the normal, for this slide, just remember the normal endothelial count is 2500 to 3000 cells per mm cube. I have already talked about pleomorphism and polymegathism. Okay, and you need to remember a painful condition associated with endothelial count reduction, and this is known as, let me write with the red, it is known as bullous keratopathy. Now, what does this bullous keratopathy literally mean? This bullous keratopathy literally means that whenever this is, suppose your endothelium, in this endothelium there is an injury and there is a big sac like this which is filled with fluid. Okay, Okay, this is like a bulla. This is a bulla. Okay, this is a bulla, and this bulle, it consists of certain fluid. Okay, it consists of certain fluid, and this mechanism is known as bullous keratopathy, and this occurs whenever the corneal endothelial count is less than 500 cells per mm square, usually at this particular count. I hope we are clear till here. Okay, now the next thing that we have to remember is whenever there is anaerobic mechanism by which the cornea is hydrated, if this occurs, that leads to an accumulation of the lactic acid, and if this lactic acid gets accumulated, it will lead to a defect in the pump mechanism. What pump mechanism? Defect in the pump mechanism of sodium potassium adipase. Okay, so this doesn't work. That will lead to, again, corneal hydration, corneal edema. So I hope I have made you clear till here. I think all the things that I've included are well clear. Now let's move on to some other topics. Now, so the cornea is transparent. I've already talked about the cornea is transparent. So what is the major reason by which it is transparent? It is transparent because there is collagen type 1, there is glycosaminoglycans, which are well arranged in the stroma. The cornea is transparent because of the sodium potassium adipase pump of endothelium, and this sodium potassium adipase pump of your endothelium leads to a dehydrated cornea, and that is also responsible for its transparency. And there is the corneal epithelium, and this corneal epithelium is also responsible for regular arrangement of the corneal epithelium, and the stromal lamellae are also responsible for the cornea being transparent. And one more thing I want to add up here is the stromal pressure. The stromal pressure is very important. The stromal pressure is much more higher than that of the intraocular pressure. The intraocular pressure, it's about 10 to 21 millimeter of Hg. More on intraocular pressure you'll study in glaucoma, but what you need to remember is this stromal pressure is much more higher. That is about 53 to 55 mm of Hg, and this difference is also responsible. This IOP difference with the stromal difference is also responsible for the cornea to be dehydrated, and for the cornea to be transparent, not dehydrated. Okay, now there are different investigations related to cornea. Okay, let's go on to the investigations. So first of all, we'll talk about investigations. I hope we are clear till here. Okay, you don't understand anything, just make your notes. Okay, just keep on writing, keep making your notes. It'll be very helpful to you. So investigations related to cornea. So what we need to remember is there is something which is known as a pentacam. So you see this regularly arranged, it's not regularly, it's steeper towards this part, but actually it's regularly, okay, circle, regularly arranged. I'll show you this picture on some other session of picture discussion on cornea. But just remember for now that this regularly arranged, this one is known as a Placido disc. Okay, this is known as a Placido disc. So I just place this Placido disc in front of someone's eye to whom I'm checking, and that transparency I'll be seeing in someone's eye. Okay, that I'll be looking. Okay, like it's like a mirror image. Okay, and if I see a steeper, if this one is, rings are closer in the steeper cornea and rings are far apart in the flatter cornea. So if the rings are steeper, that means it's closer. And what you need to remember is this is seen in keratoconus. And rings are far apart, rings are Okay, rings are far apart in the case of your flatter cornea. So F for far, F for flatter. Much more better investigation of this one is from a orb scan. Okay, orb scan. Okay, and the principle is based on the Placido's disc. So I think this much is clear. Next is pentacam. Pentacam is also very important. Okay, pentacam is also one of the investigation modality of the cornea, where we see the both the anterior and the posterior corners of the cornea. And why is it important to see anterior and the posterior? Because early changes of keratoconus. Keratoconus is a condition where your cornea is, it's there's a monsoon sign, and there's, this cornea is really thin and it goes down like this. Okay, I'll talk about this later on in the later classes. For now, just remember that this cornea, this pentacam, is responsible for looking at your both anterior and posterior part. If you see the anterior part and the posterior part, what's the advantage? Early changes of keratoconus, it occurs from the posterior part, so you can intervene early, so that the person doesn't have to go for a keratoplasty. He can just be treated initially. Okay. And you can also look into, by the help of this pentacam, you can also look into corneal guttati. Okay, this word I'll write, corneal guttati. We'll learn about this corneal guttati more whenever we talk about endothelial dysfunctions. Okay, so that's all. Now if you need to measure the thickness of the cornea, so normally, what is the normal thickness of the cornea? So 540 microns. So it's said that 540 microns is the normal average thickness of cornea. Now if this thickness is not well maintained, so in the cases like keratoconus, it will be of course thin. But what we need to remember is, if I want to check thickness of the cornea, I will be checking it with a very important instrument. Okay, I'll be checking it with a very important instrument that is known as a pachymeter. And if I have to look into the sensation of the cornea, then I can either look with a whips of cotton that you have done in the basic sciences, or you can use something which is known as an aesthesiometer. I'll be showing you the picture of this pachymeter, aesthesiometer, pentacam, or Placido's disc in other sessions of cornea. Okay, now let's go on to ulcer. Very important. So now we start the cornea topic proper. Okay, this is going to be a very long lecture, because cornea is very long topic, very long topic. People find it very difficult to understand. I too find it very difficult. Okay, so let's talk about ulcers. So in cases of ulcers, what we need to remember is, ulcers is any breach in the endothelium. It's any breach in the epithelium. And whenever we talk about ulcers, what we need to remember is, there's an epithelium, there is the stroma, there's this basement membrane, this stroma, there is this endothelium. But even though we have these many structures, bacteria, so see, bacteria, they can breach through this normal epithelium. Okay, and that is bad. That is something we don't want. And what anything that we don't want is caused by these bacterias. You can remember with the mnemonics NLLDHS. Okay, Neisseria gonorrhoeae, Neisseria meningitis, Listeria, diphtheria, Haemophilus, and Shigella. Okay, Neisseria gonorrhoeae, Neisseria meningitis, Listeria, diphtheria, Haemophilus, and Shigella. Now let's talk about keratitis. Keratitis is actually a term for the corneal inflammation. So corneal inflammation is termed as keratitis. There might be a bacterial keratitis, there might be a fungal keratitis, okay, there might be a viral keratitis. So these are the major, classification of keratitis. So I will be making a chart for you so that it's very easy for you to understand. I think you can take a screenshot. You don't need to write a lot. Okay, I'll help you out with this particular session of bacterial keratitis, fungal keratitis, and the next one being as viral keratitis. Okay, so please be with me, please write your notes. I hope this will help you. Now, so what we need to remember is in any sort of keratitis, keratitis as a whole, what is the feature that the patient presents with? It's pain, redness, photophobia, blepharospasm, discharge, blurring of vision, and there might be an ulcer formation. Very, very important, very important. If there is any sort of ulcer formation, it's better not to give steroids. If you give steroids, that might lead to a corneal perforation. Okay, so the clinical features I have already talked, and there is something which is known as a Panus formation. So if this is your eye, then what you have is you have this neovascularization around, which is termed as Panus. Okay, so most common bacteria causing keratitis, it is Staph aureus. In India, it is Staph epidermidis. Bacteria resembling fungal keratitis is Nocardia. And bacteria which causes keratitis after refractive surgeries, that means the lens laser surgeries, are actually Mycobacterium chelonae. And so there's something which is known as the ulcus serpens. Okay, there's something which is known as the ulcus serpens, and in ulcus serpens it is like a serpentine type of pattern that we see in the cornea, and this is caused by pneumococcus. Okay, there is pain, redness, photophobia, discharge, blurring of vision, and blepharospasm. So what are we going to get on examination? So the clinical features are the same as I have discussed over here: pain, photophobia, discharge, blepharospasm, blurring of vision, neovascularization. Whatever you want to write, just write it. Now, on examination, so I will make a diagram on examination features, because if you make a diagram, you'll never forget. Picture memories last longer. Okay, so you just make like this. So I'm going to make something in this circle. Okay, the first thing that I'm going to make here is, and I'm also going to make here one like this kind of picture. Okay, so please make two pictures along with me. So just make it with this one with a blue means watery means fluid means there is a stromal edema. Okay, and what you can see is a hypopyon pus, and this is known as a sterile hypopyon. In the case of fungal, I will write this as unsterile. So in bacterial, I'm writing as sterile. Now, what you can see here is, over here you can see is okay, so this is fluorescein stain positive ulcer. fluorescent stain positive ulcer. Okay, and you can see something which is known as a Descemet's membrane fold. This is a Descemet's membrane fold. So you want to make it here, you just make it like this, maybe anything like that. I just make it here, Descemet's membrane fold. And you can see something which is known as an edema. So you can just make it like this, and you can just increase the thickness and just, where does this scale come from? Okay, you can just make it, oh, I think I have to decrease the intensity, and you can just make it like this. Okay, it's well clear now. This is stromal edema. So what you can see is, you can see stromal edema, you can see sterile hyperpine, you can see Descemet membrane fold, you can see fluorescent stained positive ulcers. So this is picture memory. It helps you better, okay? So on examination, features of bacterial keratitis, if someone asks you, then you can easily write it down, okay? Next topic is, so any keratitis, it is associated with anterior uveitis. Okay, it is associated with anterior uveitis. And if it is associated with anterior uveitis, then it is of three types. Okay, uveitis, we'll be talking about uveitis later, but any sort of keratitis, any sort of bacterial keratitis, it is associated with uveitis. And it's of three types: perforating, localized, and sludging ulcer. So what do you mean by sludging ulcer? Sludging ulcer means the presence of pus, and that pus leads to a formation of a pseudocornea. Okay, so that pseudocornea can lead to an anterior staphyloma. And now what is anterior staphyloma? We'll discuss this later, okay? It's pseudocornea because that pus, it resembles a cornea, but it is not actually a cornea, but it is something which is known as a pus. Infection by virulent organisms can lead to sludging, which is continuously replaced by the inflammatory cells. That leads to pseudocornea, okay? And perforation is caused by Pseudomonas. P for perforation, P for Pseudomonas. And if that perforation occurs, then you have to go for a corneal transplant, okay? Because Pseudomonas, they have exotoxins, and that exotoxins has a collagenolytic property. Okay? And thinning causes perforation. It will not remain open as iris will seal. Okay, now this is very important. Leukoma adherens. There is this term which is known as leukoma adherens. What do you mean by leukoma adherens? Suppose there is this perforation in your cornea. Okay, then the iris over here, over here will actually seal this perforation. So if there is perforation over here, this iris will actually, it will actually seal your perforation like this. Okay, it will seal your perforation like this, and this is termed as leukoma adherens. Okay, so what is this? What it is written? It is written that thinning causes perforation. That is true. Thinning, it causes perforation. Okay, it will not remain open. It will not remain open as it is. And iris will seal this perforation. And it is termed as leukoma adherens. Now, what are the investigations that we are going to do? So, investigations. I hope we are clear till here. So you're going to write some more. So I'll just clear this. So investigations. I told you this is going to be a long lecture. Investigations. Investigations, what are we going to do is we are going to take a corneal scraping. Remember, in conjunctiva, we are using a Desmarais retractor to retract the cornea, to retract the conjunctiva to see the places. But in cornea, we are using a Cornea spatula, Cornea spatula. And with the help of this Cornea spatula, we are going to scrape the cornea. We are going to scrape the cornea. And we are going to go for a gram stain. See, gram positive negative. We can go for a meat broth. This is basically your for cooked meat broth. Okay, I've studied it in microbiology for anaerobic bacteria. Anaerobic bacteria. So we'll be talking about treatment and other features in the next video.